Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Eurofins Pharma Discovery Services, I'd like to welcome you to Accelerate Biologics and Biosimilars Development, Case Studies Implementing Robust and Simple Potency Bioassays. My name is Elizabeth Lamb, and I'll be the host and the technical director for today's event. Now I'd like to introduce our moderator for today. She is Dr. Alpana Prasad, Product Manager for Eurofins Pharma Discovery Services. Welcome, Alpana.
Hi, everyone. I would like to welcome everyone to another exciting presentation in our series of webinars focused on bioassays for biologics. As we all know, that a quantitative and robust bioassay that is reflective of the mechanism of action of the drug is a critical component of any development program. Our cell-based assay platform at Eurofins DiscoverX addresses these requirements and include simple bioassays based on the native biology of the relevant receptors for potency determination and stability testing of biological drugs. These homogeneous assays employ convenient thaw-and-use cryopreserved cells to provide excellent accuracy, precision, and reproducibility. These assays are also highly scalable and suitable for automation. Today, our panelists will be sharing qualification data for 3 ready-to-use bioassays for representative biologics as well as immuno-oncology targets. The first 2 case studies include data from the currently ongoing assay validation study at Covance UK. With that, I would like to introduce our speakers. Our first presenter is Dr. Paula Urquhart, who is the bio-CMC scientist at Covance Laboratories in Harrogate, UK. She had joined Covance about 3.5 years ago, transferring from an academic research background. She has a PhD in biochemistry from the University of Bradford. She's been focused on the optimization, qualification, and validation of bioassays for several bioassay markers, including interleukin receptors, anti-VEGF, and checkpoint modulators. Our second speaker of the day is Dr. Jane Lamadin, who is the Director of R&D at Eurofins DiscoverX. She currently oversees the development of novel cell-based assays for discovery as well as bioassays for lot release and NAB applications. Jane obtained her PhD in genetics from UC Davis and has over 18 years of experience in developing cell-based assays to support drug discovery. She primarily focused on the oncology and inflammation therapeutic areas as well as high-throughput system biology research. I would now hand over to our first presenter, Dr. Paula.
I'd like to speak to you around about the qualification of ready-to-use bioassay kits for implementation into the QC environment. So first of all, just to set the stage on why we would think about moving to a kit-based method. So obviously the established continuous culture assays are routine in cell culture, and that takes time, but also takes time not just during the week, but also during the weekend. And delays can happen. So for instance, when the cells are thawed, sometimes they don't want to expand in culture, but other times cell contamination occurs, and that delays timelines, and it makes for an uncomfortable period of time when you're trying to get things back up and running again. So those are things that we're all quite knowledgeable about. Some things that you might not know about are that pre-screening of donors in terms of primary cell cultures for appropriate levels of receptor expression can be required. And if you don't pre-screen for donor expression, then variability occurs, and if you buy, for instance, pooled cells from multiple donors, then you're looking at very wide CVs and wide variability. So these are kind of things that can be challenging when you're implementing a continuous culture into continuous culture assay into the QC environment. Another consideration is the dynamic range of continuous culture assays, and that can vary, obviously, depending on the passage, the age exactly, or the health of the cells. So whereas kits can provide with knowledge that they are produced within limited kind of passage numbers, and certainly we've got experience of where the kits are produced within passages like 3 to 5, for instance, and that kind of gives you extra confidence of reliability. So in terms of time savings, the kits come already kind of 80% optimized for your innovator or drug of interest. And then also the cryopreserved cells reduce the time from thaw to results. So these are considerations that you would think about when you're wanting to move to a kit-based bioassay. So just to then start with the first study that we're going to talk about, which is Tocilizumab. So the work scope was that Covans would test the mechanism of action kit, the IL-6 ST kit from DiscoverX Eurofins, and then feed back the performance to the Eurofins DiscoverX team, and then they would do a bit of optimization, and we would take the kit back in and assess the optimized kit for robustness. So just to kind of give you an overview of the IL-6 ST mechanism of action. So it's designed, the kit is designed to detect the binding of the interleukin-6 to the receptor with the interleukin-6 signal transduction protein. So once the agonist has activated the receptor, you have multimerization of the receptor where the enzyme fragments then combine to produce the active enzyme, and that cleaves the substrate to form light. So basically, you add interleukin-6 and you see a luminescent signal. If you add Tocilizumab, that blocks the binding of interleukin-6 and no signal is produced. And again, just to go through the bioassay outline, so you would thaw your cells. If you're looking at the agonist, so interleukin-6, and you want to assess the EC80 of the agonist, then you'd run a dose response. So you would add the agonist to your cells and incubate. If you want to introduce the inhibitor into the assay, then generally you would mix the inhibitor with the agonist, pre-incubate before you then add your add that mix to the cells, and then after incubation, read your cells on a plate reader for luminescence. So it's quite a simple outline, and it's a straightforward method to work with. So just talking around about, the comparison between the PathHunter kit and the Tocilizumab continuous culture assay, I've got a workflow comparison here just to show you. It kind of reinforces the speed difference between producing results from the kit-based method and the continuous culture method. So in the top panel here, in the top schematic, there's the PathHunter kit assay. So from plating or seeding your cells to an actual result, it's under 48 hours. Whereas when you have the continuous culture cells, you have, if you're thawing the cells to use them initially, then you have like 1 to 2 weeks of cell culture before you can then start the assay, and the assay takes 5 days. So you have, a doubling. If you're just running the assay, then you've got a doubling of time compared to the kit assay just on the assay alone. So just to get into some data now. On the left-hand graph, we have a kit-based assay with interleukin-6, an interleukin-6 dose response. So we have, in the blue curve is the DiscoverX interleukin-6 that comes with the kit, and the other curves are different vendors' interleukin-6 agonists that we tested to look for the response. As you can see from this, the interleukin-6 EC80 in the DiscoverX agonist has the lowest EC80 compared to the other IL-6 tested and gave a really good response. So the signal window that was achieved was approximately 10-fold. So all those things were very positive. When we looked at and compared using the DiscoverX interleukin-6 with the TF1 continuous cell culture, you can see that we achieved a window of approximately 2-fold. So the cells that we used were TF1, which are an erythro leukemic cell line, which is responsive to interleukin-6. So immediately you can see that we've got a nice signal window comparison between the two cultures. So then we went on and introduced the Tocilizumab inhibition into the assay. In these curves we have the Tocilizumab in blue is the reference, and we have in green and red we have 100% target potencies. So apologies if there's anybody that is red-green colorblind. The main thing that you need to understand is that they're all 100% target potencies. And then in the orange curve, we have interleukin-6 concentration dose response. So the nice thing about this data is that you can see the approximate EC80 of the interleukin-6 was used within the Tocilizumab dose response curve, and that equates the concentration here equates to the maximum signal in at the lowest point of the Tocilizumab dose responses. So the points here are a little bit top heavy, but that's something that you work with during optimization, and we worked with the EC80 of the interleukin-6 concentration and then fit our dose of Tocilizumab around that. So what you can see from that is that we would need to maybe increase the top dose of Tocilizumab. One of the nice things was that we had very nice tight replicates. That's something that in a couple of graphs I'll show you wasn't necessarily consistent when we were trialing this kit, but I'll continue with the plate layout at the moment. So part of the reoptimization that we ran was to look at using the full plate as opposed to the inner 60 wells, for instance. So what you can see if we look at the values, so what we have here actually, first of all, we have in blue the reference in rows A and E, and then samples 1, 2, and 3 in B to D and F to H. So we looked at target potencies of 50% in rows B and F, 150% in rows C and G, and 100% in rows D and H. So the potencies that we achieved were 56%, 56.1%, 147%, and 156% respectively. So for the 100% target potency, we got a value of 156. So when we looked at it, we realised that in rows D and H, if you compare those, the values are much in D and H. So the values are lower in row H compared to row D. And then when you look at the reference, it's higher than in row A than it is in row E. So you can see that we saw some edge effects. From that. So the conclusions were that we would continue with using rows B to G and keep rows A and H empty for further assays. So using the reduced plate layout in terms of we had a reference and 2 samples in duplicate on each plate. We carried on to look at more target potencies. And in plate 1, we had target potencies of 100. You can see that we've optimized the dose response curve to have more points on the lower asymptote, nice linear portion, and on the upper asymptote. And then in plate 2, we had a target potency of 50%, and sample 2 on plate 2 was a target potency of 100%. So the achieved potencies that were produced in plate 1 were 114 and 125%. So a little bit high compared to what we would necessarily want to achieve. And in plate 2, the achieved potencies were 43.3 and 173% respectively. But what I want to really bring your attention to here is the variability in the duplicates. So we did see, initially, quite a lot of variability in the duplicates, which obviously impacts the CVs, which obviously impacts then the precision of the data. So once we fed that back to Discoverix, they had optimized the kit conditions, and they qualified the kit using Actemra. So during their optimization, they used a higher cell density, and then changed some of the incubation times. So you can see that has noticeably improved the precision. So we were happy then to continue with the third aspect of this study, which was the robustness testing. So we continued by looking at the different types of incubations that happen within the assay, and we came up with a 5-factor design using Design Expert software. So we looked at the length of cell seed incubation, Tocilizumab, interleukin-6, the detection reagent 1 and detection reagent 2 incubation times. So this is the output of the Design Expert data. So in the half-normal plot here, once we analysed the data, you can see that there was no significant effect on any of the factors when you looked at them individually. But when we looked at the 2 highest impacting factors in combination, which was the detection reagent 2 incubation and the cell seed incubation, they had the higher impact, and when looked at in combination, they produced a significant result. So it's a small data set, and that might be the reason that we're seeing this significant result, but it's something that we can monitor with tracking and trending as we go on. So in the Pareto chart here, it's just another way really to show what the impact is in terms of how these 2 impact compared to the other factors. And in here, this is the analysis of those 2 factors is showing the p-value as significant. It's a bit hard to see for me. I hope you guys can all see it. And it was 0.0283. And then looking at the effects of the different factors. So in the top graph here, we've got the detection reagent 2 incubation. So at the lowest incubation time, it reduces the potency. It has a negative impact on the potency, whereas at the highest incubation time, it increases the potency. But that's converse with the cell incubation time. So at the lowest time, you have an increase in the potency, and at the highest incubation time, it reduces the potency. Oh, so one thing, sorry, that I meant to say, this is all based around looking at relative potency. All of these factors are being looked at with reference to relative potency. So there are other things that you can look at to have a more holistic approach to the design expert, which is things like looking at the EC50 or looking at the dynamic range, i.e. the signal window. So these are all types of responses that you can then look at by a design expert and see if any of your factors are impacting those outcomes. But for the purposes of this presentation, we're just talking about the effect on relative potency. So that data told us that even though we were seeing a bit of an effect with the incubation times, the 2 incubation times, if we targeted our incubations to the centre of those times, then we were hitting the target potency that we were looking for. So overall, the robustness data that we produced, bearing in mind that we had 8 runs with 2 plates per run, and all the target potencies were 100%. The overall geometric mean was 97%. So the relative bias was -3%. And then, so the relative bias is a measure of accuracy in the assay. And then the percent GSD was 14%, and that's a measure of the precision of the assay. So we would ideally have a measure of either a limit of either 15 to 20% on the precision of an assay. So this was a respectable outcome for such a small data set. Once we plugged those figures into the CPK factor, which determines how many assays you would need to run to produce a reportable result. So I'm not sure who knows the CPK statistical measurement, but basically you need to have a value of over 1, but it needs to be as close to 1 as possible. So you can see that the total was 3 in this case. So that meant that you could have 1 plate per assay. The number of assays would be 3, so that would lead to then 3 people running on one day, 3 individuals running in one day making individual preparations, or 1 person running over 3 days. So to summarise the results from the Tocilizumab Bioassay kit that we tested, we found that there was excellent accuracy with an absolute relative bias of 3%. The precision was very respectable, coming in at under 15%, and the signal window in the inhibition assay was approximately 8-fold in the kit versus 3-fold in the TF1 cells. Sorry about that typo. The next steps that we'll be carrying out will be establishment runs to test a second analyst to have a look at the variability between analysts. And so far, we've got very encouraging results from target potencies of 50 and 150%, where the achieved potencies were 51.3 and 146.5%, with the tight replicates and low CVs of under 10%. So I'd just like to wrap up with acknowledging the people that worked on this project. So in Covance, so this is wrapping up the Tocilizumab part of the presentation. The people in Covance that worked on the project were Jimmy Nuttall, Melinda Martin and Stuart Dunn. And then at Eurofins Discoverix, there was Andrew Green, Alpana Prasad and Jane Lamerdin. So before I move on to the second section, I'd just like to say that if anybody out there is working on a Tocilizumab biosimilar and would like to discuss the data that we have further, then please get in touch. We would like to be able to discuss that with you. So the second part of the presentation is the second study is the comparative study of the anti-VEGF assays. So it's a small study looking at the HUVEC, so primary cell culture proliferation assay versus the PathHunter Bevacizumab Bioassay. So again, the mechanism of action is very similar to the VEGF assay, whereby you have, the interleukin-6 assay, whereby you have the VEGF receptor, and once VEGF is bound, you dimerize, and then the active enzyme cleaves the substrate and produces luminescence. And if you have an anti-VEGF, it binds the VEGF, inhibiting binding, and then you have no signal. And the outline of the assay is very similar as well, whereas where you seed cells, you treat with an agonist to find out your EC80 agonist challenge, or introduce the inhibitor and incubate with the cells, and then read the plates once the detection agent's been added. So with the workflow comparison, again, with the PathHunter kit, you have a response, I would say, rather than saying under 24 hours, I would say within, well, yeah, actually that's fairly reasonable, within kind of 24-hour, 36-hour period. You have from thaw to result, whereas with the HUVEC cell culture, it can take 2 to 4 weeks, depending on what point you are with your primary cell culture. One of the things that you need to be aware of with the HUVECs is that they are only viable between kind of passage 1 to passage 5, so that adds another pressure within the assay time. So some of the data that we have from this study was to test VEGF 165 isoform with to get a dose response. So in blue we have the Discoverix VEGF and a different vendor's VEGF in red. So you can see from this that they actually behave quite differently. So it's quite important to monitor this and use, look at what your critical reagents are going to be within the assay. Carrying on, we use the Discoverix and VEGF as the agonist. And then on the right-hand side, we have the HUVEC comparison assay. So here we've used different cell numbers to produce the response. And that's one of the things that you need to kind of be aware of with the HUVEC or with any kind of primary or continuous culture, is that you have to start from the very beginning, whereas the kits are as I've said, optimized, about 80% optimized. So you have to start from the very beginning. So we just wanted to kind of make you aware that this is the starting point to try different cell numbers to optimize what your window and the response is going to be. So introducing Bevacizumab into the assay, we have a really nice signal window, threefold signal window from the Bioassay kit. Here it's a target potency again of 100%, and the achieved potency was 106%. With the HUVEC proliferation assay, we had an approximate 1.5-fold window. We have seen approximately 3-fold window, but as I've mentioned already, it's quite variable. And you can see immediately that the replicates here aren't as precise as they are in the kit So we carried out a study for a validation for the Bevacizumab bioassay. We used the USP guidelines 1033. We had an design where we had 2 analysts, each running 2 plates and 2 replicates per plate. And the target potencies were 50, 71, 90, 100, 121, and 150%. That meant that 6 independent runs were run per analyst to achieve an N of 8 per target potency. And this was the outcome of that data. So for every level, repeatability was calculated. More importantly, intermediate precision was calculated. So normally we would want intermediate precision of under 15 or 20%. So for this, it was a very respectable, approximately 10%. Combined within each level, it was under 15%. So we were quite happy with that, again, based on quite a small study. And the absolute relative bias was calculated per level as well, and this was found to be under 10%. So once we plugged all that data into the CPK stat analysis, we had an N of 2, so a reportable value would be made up from an N of 2, and you could have 1 assay and 2 plates per assay. So again, that showed that the assays were very accurate and quite precise. So overall, with the Bevacizumab data, we have excellent accuracy, under 10% in individual levels, a respectable precision, under 15% per level, and from the small data set, the results showed that we could run 2 plates for 1 assay to produce a reportable result with an N of 2, and that the dynamic range is wider with PathHunter assays compared to the primary assay. At the moment, we're looking at a bridging study between the HUVEC and the kit-based assay, because that's something that regulators are very interested in. And I'm sure we'll be updating with that kind of assay, hopefully at conferences to come. So I'd like to acknowledge again the people that have worked on this project, who are Paul Caldwell, Emma Bowen, James Hunt from Covans, and at Eurofins, Jane Lamerden. So the overall conclusions are that the key points really are that the assay must work straight from the box, because you don't have the opportunity to optimize all of the factors that you might want to in terms of cell number, etc. You need it to deliver a consistent performance with each lot, and you have to test that, you have to qualify basically the critical parts of that between kits. You don't want any anxiety with kit reagents, so for instance, volumes of detection reagents, etc. And you want it to be easy to transfer to other sites, and you want it to be comparable to the continuous culture method. So you really require a bridging study to show that before you can then take these kind of kits through validation and use them for filings for innovator or biosimilar type molecules. And then critical reagents, obviously you need to know that you've got lot-to-lot reproducibility, and that you can ensure a long-term supply, and that your cell banks the cell banks that you will receive will be viable for that long term. But overall, the advantages of using the kit base that we have demonstrated, hopefully, is that they're very fast from thaw to result. You don't need any cell culture in terms of routine cell culture equipment. they're reproducible, they're accurate, and they're simple to use, and there's very little in-house technology that's needed in terms of developing reporter cell lines, etc. So I will stop there, and I'll pass over to my to Jane from Eurofins DiscoverX. Thank you.
Thank you, Paula. That was really great. I appreciate having the opportunity to speak to everyone today. I'm going to change gears just a little bit now. So what I will be covering in the few minutes remaining in this webinar is some development that we've done for a PD-1 signaling bioassay kit that comes from our immune-onc menu. As many of you know, there's a significant amount of excitement about the success of checkpoint inhibitors, particularly modulators of PD-1 and CTLA-4. And with that enthusiasm, there is a lot of interest in developing therapeutic agents against other T-cell relevant receptors, both the co-inhibitory receptors and co-stimulatory receptors. As you're all aware, the co-inhibitory receptors are therapeutic targets for blocking antibodies to enhance T-cell stimulation, while agonist antibodies to the co-stimulatory receptors also act by stimulating T-cells in cancers. So we have developed a number of assays against the targets that are shown here, both as signaling assays and also to support development of bispecific antibodies. But what I'm going to focus on in the next few moments is a bioassay that we've developed for PD-1. The assay shown here is the mechanism of action of PD-1 is shown here on the left. And so what we're focusing on is a very early event in the activation of the PD-1 receptor. So what we're looking at here is a very early event in the activation of the PD-1 signaling receptor. So PD-1 itself is expressed on the surface of T cells. And when it engages its ligand presented on the surface of the APC or tumor cells, an intracellular event occurs where kinases phosphorylate these ITIM or switch motifs in the C-terminal tail of PD-1, which results in recruitment of SHP phosphatases. So this particular, the SHP phosphatases then dephosphorylate the TCR and CD28, resulting in downregulation of T-cell signaling. So what we've done here is designed an assay that captures this early event in the T-cell signaling, PD-1 activation. So what we're showing here on the right is the assay that we've developed, which is a co-culture model where we are expressing, we have GERCAT cells that are expressing PD-1, which is full length, using our beta-gal complementation system where PD-1 is fused to the small fragment of our beta-gal reporter, whereas the SHP phosphatase is fused to the larger beta-gal reporter. And when these two are co-expressed in the GERCAT cells and then co-cultured with U2OS cells presenting our ligand, we're able to see an increase in a signal due to complementation of the two fragments. When we treat those cells with either an anti-PD-1 or an anti-PD-L1 molecule, what we see is inhibition of that signal. So this is an assay that is a simple homogeneous assay where we're able to run this assay in less than 6 hours. We simply plate the GERCAT signaling cells in a plate, add the anti-PD-1 antibody, incubate for an hour, and then add the PD-L1 or PD-L2 presenting cells, incubate for an additional hour, and then add our detection reagents. And then 3 hours later, we read that on a benchtop luminometer. So it's a very robust assay. And here on the left, we're showing data that demonstrates its specificity for its ligand. So here we're showing data with PD-L2. The dark curve here corresponds to PD-L2. and stimulation or co-incubation with ligand cells presenting an unrelated receptor or ligand, HBEM, shown here in gray, has no effect on the assay. Similarly, if we look at the specificity for an antibody in the assay, we see that an anti-PD-1 assay is able to inhibit the assay robustly, while an anti-TIM3 antibody shown here in the teal curve is not able to inhibit the assay. And finally, what we can see here on the far right is that this assay responds robustly to both of the currently marketed anti-PD-1 therapeutics, pembrolizumab and nivolumab. So what we've done now is taken these particular therapeutic agents and used them to qualify this PD-1 signaling bioassay. And some results here, representative results from the qualification, are shown in these graphs here at the top. These are data from PLA, where we're assessing the, in this particular case, pembrolizumab at 4 different concentrations. From the left to the right, it's 50 versus 75, 125, or 150% concentrations of pembrolizumab relative to a 100% reference standard. And what you can see from these data is that qualification, which was performed with 2 analysts in at least 4 experiments per concentration, results in very good accuracy, precision, and linearity, not only for pembrolizumab, but also for nivolumab, as shown here in this chart. And both therapeutic molecules demonstrate excellent dilutional linearity, values of greater than 0.995. Importantly, if we test this assay with a forced degradation sample, in this particular case, it was degraded at 65 degrees for 5 minutes, you can see a robust shift in potency relative to our reference standard, again indicating that the assay is stability indicating. So we have developed a number of assays now that are off-the-shelf bioassays targeting a number of therapeutic molecules, and these can be used either for innovator drugs or as assays for development of biosimilars. So shown here on the far left are the qualified bioassays that have been qualified with the named drug that you see there. And on the right, we have a list of assays that are currently in development. So we have cell lines that are responsive to these particular drugs, and the assay-ready format that Paula described to you and we just talked about for PD-1, that format will shortly become available. And additionally, we have a number of other target assays that are MOA-based, targeting receptors of interest for development of a number of therapeutic agents. And these are just some examples of ones that are available as off-the-shelf, but have not yet been qualified with an innovator or a client molecule. But they are available as assay-ready. Finally, we have a number of other target bioassays that have been recently developed and will soon reach our product catalog. And if you're interested in any of these, I would suggest that you contact us and we can provide some details for you. So I just wanted to conclude with if you have interest in any of the assays that have been described here, you can reach us at Eurofins Pharma Discovery at either our technical support or on the web. You can see our list of bioassays, and then of course we can respond to any questions that you may now have.
Thank you, Jane, and thank you, Paula, for the data sharing that both on the different bioassays. I'm going to go through some of the questions that have been sent in through the chat panel, and any of the additional questions that you may have, please keep sending and we will respond to you via email. So for the very first question, Paula, the question is, could you elaborate on the continuous culture TF1 assay and what is the readout for this assay?
Yeah, so the TF1 assay is a 5-day proliferation assay, and the the readout is fluorescence, so I think it was Alamar blue that was the endpoint.
All right. Okay, thank you, Paula. There's another question for you. For the Tocilizumab bioassay kit, what degree of change in the cell incubation time produced the difference in the DOE with the design expert? I'm not going to go into the actual incubation times that we looked at, but what I would say was that the incubation, I mean, the data that we've got from that was it was from a small data set, so I'm not absolutely confident that the effect that we see by using the design expert is actually a true result, and it's something that we would want to monitor as the more data that we get. But really, what I would say is that the potencies that we saw were between about 100, say, kind of 85 to 120%. So ideally, what we would want to see is from 90 to 110%. So we would initially we would keep any criteria that we had around the potency. We would keep it wide initially, and then tailor it down once we got a bigger data set. All right. Thanks, Paula. Another question is, how do you ship the cells, these frozen vials? So the cells are shipped as a part of the kit. These are cryopreserved cells and are shipped on dry ice, and upon receipt must be stored in the vapor phase of the liquid nitrogen. So another question is for Paula, for the pre-screening of the donors that you had mentioned, how often do you need to screen the donors, and how many donors do you screen before you combine them in a pool, and what are the different parameters that you're looking at? Ideally, we would screen perhaps 2 to 3 donors, but we wouldn't pool them. We would only use those single donor cells. So the kind of parameters are the parameters that I've discussed in terms of signal window, response to the agonist, and cell number. Yeah, we wouldn't pool those. We wouldn't pool them. We would just use them as independent donors. And I guess it's then you've got to make sure that wherever you're getting those cells from have enough cells for you to then carry out the tests that you need. But it's a continuous thing. It's when you're talking about primary cell culture, it's a continuous process. Thank you, Paula. This question is for Jane. What is the difference between your PD-1 bioassay and Promega PD-1 kit?
Well, so the first difference is that we are looking at an early event in the activation of the receptor. So we're looking at recruitment of a protein to the C-terminal tail of the receptor. So this occurs very rapidly, whereas the Promega kit is looking at activation of, I believe, an NFAT reporter. So it's a transcriptional event that occurs downstream. Secondly, the Promega kit requires activation of the TCR in order to detect the inhibition by PD-1. Our assay is looking at PD-1 in isolation, so it does not require any TCR activation. So those are the 2 primary differences.
All right, thank you, Jane.
I'd like to thank our presenters for today, Dr. Jane Lamerton, Dr. Alpana Prasad, and Dr. Paula Urquhart. And I'd like to thank the folks at Eurofins for sponsoring. Most of all, I'd like to thank those of you who came and spent this time with us. We know you're very busy, and we're very grateful for the time that you spent getting some answers to your research questions. So from all of us at CHI and Eurofins, thank you so much, and have a great day.